welcome back to the Incubator at the Bench, the neonatology physician scientist podcast, where we're focusing on basic science research related to neonatal health. My name's David McCulley. I'm a neonatologist and a physician scientist at the University of California in San Diego, and I'm very lucky to be able to co-host this podcast with Dr. Betsy Crouch. Betsy, would you mind introducing yourself and our guest for today?
Happy to. Hi, everyone. I'm Betsy Crouch. I'm an assistant professor at the University of California, north from David in San Francisco. I am a neuroscientist, a vascular biologist, and a neonatologist, as you all are aware. I'm very pleased to introduce today my neighbor, my very distinguished neighbor, who's also a good friend, Dr. Tippi MacKenzie. Dr. MacKenzie is a professor of surgery and the Benioff UCSF professor in children's health here at UCSF. She's also the John G. Bose Distinguished Professor in Stem Cell and Tissue Biology. And recently, in the past few years, was elected as the director of the Broad Stem Cell Center for the Institute of Regenerative Medicine here at UCSF. And we have this very cool building that sits on the hill here at Mount Sutro. In addition, she was recently elected to the National Academy of Medicine. Thank you so much, Dr. MacKenzie, for being here with us today, and we're excited to talk to you.
Thank you so much, Betsy and David, for inviting me. I'm excited to have this conversation.
Hippie, could you start just by talking a little bit more about what your research focuses on and also what you do clinically? I think it's really exciting for our audience to hear kind of how we've built these careers in studying the questions that we study, but also what we do as clinicians.
Thank you. Yeah, so my lab studies maternal fetal biology to understand disease onset and use that knowledge to develop therapies for patients with pregnancy complications and genetic diseases in the prenatal period. I'm trained as a surgeon. I trained as a pediatric and fetal surgeon, and I've been at UCSF for 18 years. This was my first job after fellowship. And for 14 years, I, took call as a pediatric surgeon. I, worked at the bedside with both of you. And, I love taking care of the babies, and I think it's an amazing relationship between surgeons and neonatologists to take care of these really vulnerable infants. So 4 years ago, as Betsy said, when I became director of the Stem Cell Center, it just became too much to both have a surgical career and run a lab and run an institute. So for the past 4 years, I've stepped away from doing surgery. And so my clinical life right now is my clinical trials of new prenatal therapies.
Yeah, we have so much to jump in and talk about. I'll just say that, sort of recognizing these early moments that Tippi and I together put my first baby on ECMO as when I was an attending who, had a good outcome. And, it's these, moments that you have with your friends and colleagues that where you are really united in this common goal and you achieve that goal. And then you look at each other and think, and how's your science going? After everything, after the dust is settled at 2 a.m. So I'm really grateful for, to both of you for being able to have those, clinical and scientific moments back to back sometimes.
Yeah, those are magic times for
They are
sure.
magic times, I think. And, one of the perks of this job, right, is that like there are these moments of inspiration that you get to kind of captivate on in the middle of the night you know, when progress can be made and it feels good.
Yeah, it is so pure. Like we're just all working towards saving a child's life.
Hmm.
And yeah, that's really magical.
Yeah. 24/7 often, right? So, yeah.
From many different approaches using a lot of different skill sets. I was wondering about that, Tippy. What could you describe a little bit like what inspired you to focus on the questions that you're focusing on now and the research training that you had early in your career that allowed you to build your research interest?
Yeah, thanks for the question. So my path to clinician scientist mode was a pure MD path. So I didn't take the time to get a PhD in medical school or before or after. But I'm really lucky that I've had amazing mentors that have, taught me to do science along the way so that I could be successful running my own lab. So to your listeners or viewers out there, you can be a clinician scientist if you haven't gone through an MD PhD program. You just have to be, really focused and um, intentional about how you get that training. So in medical school, I was really lucky to work in the lab of Dr. Robert Sapolsky, who's an endocrinologist, anthropologist, biologist, just one of the most amazing human beings that I have worked with. And I got to so Stanford has an interesting program where you can do your first 2 years and 3 years and do a lot of focused research. And I learned in that lab just the love of science and the joy of discovery, like just asking questions. It's really he does curiosity based research that has ended up translating. Um, and that was a really strong foundation. And then when I went to surgery residency, as you guys know, for surgery residency, everybody takes out a couple of years to do some scientific research. And I should say, at the end of medical school, when I was doing my rotations, on my surgery rotation, I got to hear that this whole field of fetal surgery existed. So, of course, it was invented at UCSF in the 1980s. And so this was, I'm going to give away my age. This was 1996. I was a surgery resident. It was my second to last year, 4 out of 5. And I thought to myself, wow, that sounds so interesting. And, with the freedom of being a student, I could just call the UCSF, Division of Pediatric Surgery. It was before email, so somebody answered my call and was super helpful. And within a month, I was the nerdy sub-I on Dr. Michael Harrison's surgery service. And so he, as you guys know, has been a pediatric surgeon here for many years and is widely regarded as the father of fetal surgery. So I got to stand in a corner when Dr. Harrison did a a sacrococcygeal teratoma resection on a second trimester patient, and it was just the most incredible thing I've seen. I mean, talk about magic. 20 people around the room, just trying to take care of these two patients, the mother and the baby, and the surgery was a success. And you can imagine, I was left thinking, oh my God, like that's literally what I want to do when I grow up. And then a couple months later, I saw an article in the New England Journal that was the world's first in utero stem cell transplant in a pregnancy with severe combined immunodeficiency, and so that was done by Dr. Alan Flake and colleagues. And so all of these things kind of came together, and I entered surgery residency thinking, A, I want to be a pediatric surgeon, and B, I want to, go figure out some of this, stem cell transplantation stuff, because you could tell even then that that was going to be the real future, right? So there's the anatomic malformations that you can treat with surgery. But there are so many more genetic conditions that can benefit from scientific input into this field of surgery and obstetrics and gynecology. So then I was lucky to take 3 years out to do research in Dr. Flake's lab. He was at CHOP by then, and really got a good foundation of the flow cytometry and doing animal experiments and some of the other skills that you need, so that when I finally then finished my surgery training and then my pediatric surgery fellowship, I ended up going to CHOP again. I was set up to start my lab here at UCSF.
That is so exciting to hear. I mean, you have been inspired by so many great people that, whose research I've followed so closely just because I have a similar interests. And it's, I think, so empowering to have like the exposure that you had to be able to connect with people who are, at the cutting edge of fetal surgery, at the cutting edge of prenatal treatment. So that it is so exciting. I just because we've been really interested in like how to enable more people to take this path of being a physician scientist. You mentioned that program at Stanford where you said you had more time to be able to focus on a research path. Could you just talk about that a little bit? Like, did you go in thinking that you might do that, or how did you get introduced to that program?
It's just how Stanford Medical School works. I don't know if that's still how it works, but the most amazing part about it is you only pay for 4 years of medical school, and then but you can take as long as you want to finish. And so the majority of my classmates took 5 years, and the usual breakdown of that was 3 years to do your coursework, and then the standard 2 years to do your rotations. And so it also created an environment where basically everyone around you was doing some kind of research. And it's different kinds of research, right? Clinical research, bench research, global health research. And we were all, and then you also, because everyone is taking the 2 years and 3 years, you're not limited to your class. Your class is filled with first, second, and third year students because everybody is doing a different combination of when they take immunology or genetics. So it created an amazing environment. I'm sure there are other medical schools who do that, but it was a really special part of Stanford that made your MD feel like an MD plus.
Yeah, I think that's a great program to hear about, and I think for folks who are actually pre-medical school that there are a number of, I think, more open-minded ways to consider medical school these days, thinking about the way that we practice medicine now, which is not that you have to memorize a bunch of things that are in a textbook, but that you have to know how to investigate a problem. Now, know how, to form a differential diagnosis about the patient who's in front of you, and then also know how to push the field forward. So to gain those 2 different skill sets, yeah, I think there's been a number of educational initiatives. I'll just say one other program that I'm familiar with is that at Columbia, they have a 3-year MD after getting a PhD. So for folks who, thought about being a scientist first and then decided that they wanted to go to medical school. I have a clinical fellow in my lab now who's an adult gastroenterologist who really, really tried hard not to go to medical school and wanted to be an educator. So she did a PhD first and then realized that she could not that really taking care of people and their medical issues was her passion. And so then she went and did the 3-year program at Columbia. So yeah, there are, I think, a number of other ways to conceptualize the traditional 2 and 2 years of medical school these days.
And the other thing that's striking to me is in this day and age, there's knowledge is vast and growing at lightning speed. And so to think that any one person um, can obtain the training they need during their years of education to be successful, no matter where the science and patient questions lead you, I think is a bit naive. So I'm always just thinking, do I have the training? When a scientific question comes up or when a patient problem comes up, it's really important to think, do I personally have the training to do this, or do I need to phone a friend? And as Betsy knows, I'm always thinking about, for a scientific question, who can we collaborate with? Who's got something to bring to the conversation? Because even if everybody around you has amazing training, they're all MD PhDs, they all look at problems a little bit differently. And I think it's a wonderful thing to consider, just working collaboratively, as Betsy and I have recently done.
Yeah, maybe that's a nice segue to sort of, getting into your research now. I was, telling Tippi before we were on air that, even though we're neighbors, it was a great opportunity over the weekend to really, dive deep into her papers and her, the work coming out of the lab because I'm very familiar to honor her for a second about the way that she enables her trainees. There's a number of trainees who have been first author on these prominent papers that are now, going on to put in a bunch of applications that I've been fortunate to support too. But, if you could speak a little bit about the research that you're, currently investigating in the lab and where you see that going in the future, that's something that I'm very excited to talk about.
Thanks, Betsy. So, the lab really works broadly on understanding fetal biology so that we can treat genetic diseases in patients before birth. There's an enormous opportunity right now because of the convergence of three things, right? So, I mentioned Dr. Harrison invented fetal surgery in the 1980s, and now there are fetal treatment centers all over the world. um, Prenatal diagnosis. It used to be you would just go get an ultrasound and, we would look at, are there 10 fingers and 10 toes? Now, ultrasound, MRI, everything, of course, has become more sophisticated, but there's also genetic screening of, parents are getting carrier testing before they conceive, and there's also more genetic testing even in, mother's blood in the first trimester of pregnancy. Um, and so there's an opportunity to diagnose genetic diseases that might not have any manifestation on the regular ultrasound screening. And then with the invention of CRISPR by our UC Berkeley colleague, Dr. Jennifer Doudna, and um, the additional inventions like base editing and prime editing by Dr. David Liu at Harvard, there is an amazing momentum to treat genetic diseases with their root cause. And I'm really excited to put all of those things together so that we can develop programs to provide genomic therapies for patients before birth.
That's very exciting. I mean, I have recognized the same convergence of those, 3 things as being an incredible opportunity, and it's amazing that you've been able to focus on it and really try to, push it forward and try to get it ready for, clinical trials, which it sounds like you're working on already. Are there Was there a particular condition you started focusing on, or was it just thinking about, like, "Okay, what are the conditions where we could apply these approaches?" How did you think about it, and what's your approach right now?
Thanks for asking. So there are a number of modalities that you can use to treat genetic diseases. So the first one is the stem cell transplantation that I had read about when I was a medical student, and still when I started my lab, I wanted to sort of move down that path and work on stem cell transplantation. And the disease we started with there is alpha thalassemia major, which is a condition where the alpha globin genes are either mutated or deleted. And patients get very sick before birth, usually in the second trimester, and they can be treated with in utero transfusions. And initially we thought, well, maybe we can treat them with stem cell transplants. It turns out the biology isn't quite feasible for that because there's so much hypoxia in the fetus. And so now we're working on a gene therapy and genome editing approach for that. So alpha thalassemia sort of came to top because you diagnose it before birth because there's an ultrasound finding. The fetuses develop fluid collections around the heart, around the liver. And it's also very compelling because, the pregnancy doesn't survive unless you do something like give blood transfusions. And then there's an opportunity to do blood transfusions plus something else. And from there, we've moved on to other genetic diseases. So right now we have a clinical trial where we're giving enzyme replacement therapy to fetuses who have lysosomal storage diseases. Again, some of whom present with that hydrops finding of fluid collections around the liver, usually, but also some who don't have any ultrasound findings, but we know from research for decades that there is onset of organ damage, often irreversible organ damage before birth. So the genomic approaches really have been tested only in animal models, and there hasn't been a human clinical trial of a genomic approach, either a gene therapy or genome editing. Because of course that's we need to make sure that we're involving ethicists and patient advocacy groups, and really making sure that the genetic therapies we implement before birth are not going to be passed down over generations. And so there's a lot of work done in my lab and those of colleagues who are in this area to make sure that we're moving responsibly towards what might be a definitive cure for these patients.
That is how I think about your platform, which is that you have currently, maybe, these things will change, but you're thinking about stem cell therapies, and then you're thinking about genetic or genomic therapies. And I guess to go back to the stem cell therapies, I think it can be educational to hear a little bit about some of the challenges that we've encountered. So do you mind telling us a little bit more about the biology of the stem cell therapy for alpha thalassemia and the lessons you've learned from that experience?
Yeah, of course. So it sort of goes back to that first report that I had read as a medical student, where a child with severe combined immunodeficiency was treated successfully. And after that, there was a lot of excitement in the field, and people came to realize that it really only works in case of immunodeficiency. And that never made a lot of sense because, the fetal immune system is supposed to be immature. So is it the fetal immune system that's then rejecting the cells that you're putting in these other instances, or is it something else? And so when I started my lab, a fascinating set of papers came out from Mike McCune's lab where they demonstrated that during development, fetuses develop regulatory T cells that recognize and protect against an immune response to their mother. And this happens because cells cross the placenta back and forth. And so you can imagine as you're training and educating the fetal immune system, you want to train it to recognize self and not reject its own self. And these maternal cells that have been crossing into the fetus, are sort of seen as self in a way, and developing the same kind of regulatory T cells, as well as in a mouse study we showed that they're deleting T cells that are reactive against cells that are encountered during pregnancy. And so that was a good reason to think, okay, well, if there are regulatory T cells against the maternal antigens, maybe we should be transplanting maternal cells. And sort of looking back at the literature, people had not been transplanting maternal cells. Um, and then there was another study, actually, the first paper out of my lab, where we did a mouse study where we showed that in mice, at least, the maternal immune system sort of coming in as a result of this trafficking, more cells come in, and actually maternal T cells come in, and they mediate rejection of other cells that you would have transplanted into fetuses. So that was sort of a second reason to transplant maternal cells. And so that in the hubris of my youth, I was like, "Oh, I solved this, decades-long problem. This is why the patients weren't engrafting. All we have to do is transplant maternal cells." And, at the same time, Dr. Flake's lab had actually, they were on the same path, and they did an experiment in dogs where they transplanted maternal cells. Lo and behold, the dogs engrafted. They were tolerant to their moms. They got kidney transplants from their moms. So all this was really exciting. And I wrote an investigational new drug proposal to the FDA to do the first, clinical trial, because a true clinical trial hadn't been done in the field. And I chose alpha thalassemia in conversation both with the FDA and with, the set of colleagues around the world who care about this. I actually started a little society called iFetus so that we could all sort of talk to each other, so that everybody wasn't doing like tooth transplants for, this disease and three transplants for this other disease, different cell sources, et cetera. Um, so we decided to go for alpha fetuses with alpha thalassemia because they are so sick, and there's this, also ethical rationale to try to do something. And so we did this trial, and it was, it's an amazingly rewarding part of my career, because these patients, they didn't have any other options. They weren't even being given transfusions. Um, so we ended up doing a lot of outreach and public health, and talking to obstetricians like really all over the world, and, forming an international consortium. And we ended up treating 6 patients, and we just didn't get engraftment. We got very low levels of engraftment, not at all what we would have predicted based on the cell dose and what, the dog study had shown. And then looking at the data more carefully, it seems like in the hypoxic environment of those fetuses, the bone marrow is so revved up, there's an expansion of red blood cell precursors and hematopoietic stem cells. So if you sort of think about your donor cells playing musical chairs in the bone marrow, or, with the host cells, because we're not doing any conditioning to wipe out the host bone marrow hematopoietic stem cells, it really, it might have been ethically the right disease, but it was biologically absolutely the worst disease, probably. And so, I mean, I think it's a good lesson in sort of like you got to pick yourself up and figure out what you did wrong and what you're going to do moving forward. So for alpha thalassemia, we're, all systems go in developing a gene therapy approach, because that should work. And again, collaborating with Don Cohen at UCLA, who's developed lentiviral gene therapy approaches, as well as Kyle Cromer at UCSF, who's developing a genome editing approach, to really try to figure out a different solution for these families, because we have come to realize there are a lot of patients affected with alpha thalassemia and they have no curative options. And then for the stem cell transplant, really thinking about, okay, if alpha thal was the worst possible disease, what's the best possible disease? And that I think is Fanconi anemia, where it's a DNA repair a defect, and so there should be a survival advantage to wild type hematopoietic stem cells. And so, collaborating with Anishka Chekowitz at Stanford, who's an expert on Fanconi anemia, and a close colleague and friend. So she now is leading this with we're just helping. And so she did a fetal mouse study where she showed that when you transplant a fetal mouse, the level of engraftment goes up over time. So there's a proof of concept there. And she just got an IND to do a phase 1 trial of in utero stem cell transplantation in Fanconi anemia.
Woohoo!
You're like you got to resilience.
No, bravo, right? 'Cause I think that is the takeaway from this story, which is that the biology is telling you something, and it's telling you where your next step should be, even if the initial disappointment is not that the next step is that, check, we're done. Check, we figured it out. So in fact, maybe, I mean, still more work to do on alpha thal, but maybe for Fanconi's anemia, it enabled, maybe a more faster acceleration of Anishka's study, in that disease.
I hope so. Yeah, I'm really excited about that trial. Yeah.
Just because we have kind of a broad audience, I was wondering, could you be even more clear about, like, how the stem cells are given, and how you know how they're engrafted or if they're not engrafted? Like,
That's awesome. Yeah, I wanted to talk a little bit about delivery as well, because I think that's also a really interesting problem when you have 2 patients, as we think about the prenatal baby.
Of course. Yeah. So the delivery, I should preface. So Dr. Harrison started doing fetal operations in the 1980s. But if you go back to the 1960s, people started giving blood transfusions to fetuses through the umbilical vein for a really common condition called Rh disease, right? If mom's Rh negative, baby's Rh positive, mom can develop antibodies after the first pregnancy. Those antibodies go in and chew up the red blood cells of the baby, and so you can give blood transfusions. And this is a really common procedure that's performed all over the world. Obviously, it's technically delicate, but lots of talented maternal fetal medicine doctors out there doing it. It's ultrasound based, so it's just little local numbing medicine on mom's belly, and then there's a longish needle that finds the umbilical vein and injects blood. And so you can imagine that if you can inject red blood cell transfusions, then you can inject any of these medicines. And so that's how the stem cells are delivered. So in our clinical trial and in the upcoming Fanconi anemia clinical trial, we're taking bone marrow stem cells from the mom. So, it is an involved procedure. So, under the epidural anesthesia that you would get for childbirth, we do a standard, iliac crest bone marrow harvest with the pediatric bone marrow transplant team. And then the cells are processed in the pediatric bone marrow transplant lab where they, get the CD34 fraction and then, spin it down to a very small volume because the fetus is tiny. Um, and then we infuse them under ultrasound guidance. So that whole process takes about 8 hours, and it's always an amazingly exciting day with just a team of about 50 people touching um, everything along the way. Um, and so that's how the stem cell transplants are done. That's also how the enzyme replacement therapy clinical trial is done, where we infuse the recombinant enzyme for the specific disease. And that's how I imagine a gene therapy or genome editing approach would be done. So we just submitted an investigational new drug application for an AAV gene therapy for a severe genetic disease called GM1. So the delivery isn't really the problem. It's the understanding of the biology and choosing the right patient population and really making sure we're working with the patients to figure out what would be a reasonable kind of a therapeutic approach to then deliver.
that's great. And then the other question is just how, I mean, it's one thing to do these studies in mice where you can have a fluorescently labeled cell that we can follow and things like that. But when you're thinking about in a patient and in a clinical trial, how do you think about that? How do you know that you have engraftment or be able to follow the progress of the stem cells once they've been injected?
Yes. Oh, yeah. Great question. So thankfully, there we can use the CLIA certified lab tests that are used in the bone marrow transplant field, where they are, looking at genetic differences between the donor and the recipient, using PCR, to give percentages of engraftment. So, so in the 6 patients in the clinical trial, so we did 2 different kinds of tests because that clinical test is accurate and it's CLIA certified, meaning you can report back results to the patient. It's only sensitive to about half a percentage point of engraftment, which of course, if you didn't engraft that half a percent, it didn't really do you any good. But in terms of the research method, we can actually go even more sensitive, and so for the clinical trial patients, we were also, taking some blood for the research part of the test and doing flow cytometry to isolate specific cell populations and doing quantitative PCR on those specific cell populations. And then we could read out lower percentages of those of mother cells in the baby's blood. And we did that throughout a year. And then we did some other research studies looking at, well, even if we had low engraftment, did we actually have tolerance to the maternal cells? Because another part of the study, which I didn't mention, is that you don't really have to have a fully, I engrafted 10%, 20%, and therefore this is curative. You can actually have, maybe you engraft about half a percent or 1%, but then that tolerance to the mom that the baby has before birth, if you continue to have some level of maternal cells, you can continue to make regulatory T cells to sustain that tolerance, and then you can do a booster transplant without all the immunosuppressive part of the conditioning that you did that you usually do. So, in 2 of the patients, we actually did see tolerance to maternal cells, even though their levels of engraftment were not in the curative range. And in fact, for one of the patients, we got ready to do that booster transplant. But then the family didn't want to have another transplant. They were very happy with getting chronic once a month transfusions for their alpha thalassemia major. So we weren't able to test that portion, sort of part B of this protocol. But yeah, so there are a lot of these research tests that you can also bring to play.
Is there also a way that, I mean, as we know, the definitive therapy would be a bone marrow transplant for the patient with the alpha thalassemia major. But is there a way that like the prenatal transplant could make that process better or less, as we know, it's a dangerous procedure. I mean, all things being considered, that there are many patients who undergo this, thankfully, without any complications. But, have there been any insights into improving the current, curative standard of care?
Right. So great question. So the current curative standard of care, as you said, it can be dangerous mainly because you have to do the conditioning of the bone marrow. And so there's for your viewers or listeners, so there's 2 kinds of medicines you have to give to do a bone marrow transplant. One sort of wipes out the bone marrow so that there's space for your new cells. So you wipe out the person's own hematopoietic stem cells in the bone marrow so that there's space. And then the second is you have to immunosuppress. So you have to wipe out their T cells and B cells so that they don't reject the new cells coming in. So we're hoping, and neither of those is safe enough to do in the fetus. So what we were hoping for the fetal transplant is you just give a lot of cells. So even if you haven't wiped out the bone marrow, as they're playing musical chairs, they're going to sort of outcompete the host cells. So we give a lot more cells than you would in a regular bone marrow transplant. And then you're transplanting from a donor that they're already tolerant to, so you shouldn't have to give the immunosuppressive conditioning. So that's the hope. And what we're hoping for with the in utero transplant is even if it doesn't fully engraft, then after birth, you might still have to do that conditioning portion where you wipe out the bone marrow, but you don't have to do the immunosuppressive part of the conditioning, um, because they're already tolerant to their mom or the donor that you've used. Um, in terms of how to do, if they haven't had fetal therapy, how to do a regular bone marrow transplant safer. I think one of the things that's been helpful is just shining a light on this long neglected disease and asking all of our colleagues around the world, how many patients with alpha thalassemia have you transplanted? What protocol did you use? Are there things we can learn from each other, etc.?
Yeah, thank you for that. I mean, I'll say that I get the benefit of walking in every day as I walk to my office. There are 2 sort of, artistic pieces in the hallway that are present as I walk to my office, and one is Tippi has a map across the world of her patients who've enrolled in her clinical trials with little, push pins, and I think it's really exciting, um, to see, where these patients are coming from, how they're hearing about this exciting research, to feel like, you know, we're, you know, to be to be honest about it, like we're kind of a beacon of hope for certain, long neglected diseases. Um, and then there's a picture of all of these babies or children who have been treated, and so it's quite inspiring, I think, just to remember that like the world is a big place and that there are, collaborators, and I say that in terms of the scientists, but also the patients and the families who Tippy's done a lot of great work, in collaborating with the patients and the families and understanding what are their goals, and then in designing her both basic science and clinical trials, towards the most important collaborators potentially, which is the patients. So, yeah, so I'm grateful to you for putting those, mementos and those signs up to remind us, how we do our science matters. So thanks. There's so many exciting directions to go to in our last couple of minutes. Do we want to talk about, maybe industry collaborators, I think is an exciting direction that our listeners would appreciate. I think getting some tips or, tips from you and your experience.
Yeah, thanks for the question. So we've had some really fruitful collaborations with industry for testing some of the products that have been developed for patients after birth to determine whether or not they would be safe and effective to use before birth. So, for example, the lysosomal storage disease in utero clinical trial I mentioned, you can't do a clinical trial, of course, without doing some of the initial animal studies. So we did a mouse study in collaboration with the industry partner who makes the postnatal enzyme for. In this case, we use MPS7, which is known as fly syndrome. So that was a really nice collaboration. And from that, we were able to obtain data about the safety and efficacy for in utero enzyme replacement therapy so that we could present that to the FDA. We worked with industry partners to test various gene therapies, AAV gene therapies, and right now we're doing a lot of work in lipid nanoparticle delivery of genome editors to in mouse models, prenatally in mouse models. We've also worked on antisense oligonucleotides again with the lead industry partners there to determine whether the product is safe and effective even in a large animal study. Um, so I think it's broadly speaking, it's hard to think about developing a therapy like purely academically to then take that all the way to the patient, and that's where industry partnerships and collaborations can be so helpful. So I've been really fortunate to be able to participate in those.
And how do you meet your industry partners? You put out an ad on,
No, a great question. In some cases, at conferences, probably or through word of mouth or through other academic collaborators. Um, there have been a couple instances where I sort of cold emailed somebody, and I think, I mean, it's compelling to want to develop a treatment for patients before birth. And so I think we're really we all have the same goal, right? We all have the goal of helping human beings. And and I think that, people in general want to work together. So,
Yeah, cheers to that. I think it's compelling to want to cure, right? To truly cure diseases and to give a child a trajectory that they never would have had before when the opportunity is there prenatally that might not be there postnatally.
exactly, yeah. So the biological opportunity.
I do think that's one of the most exciting things about our job is that as a physician scientist, we get to help promote these bridges that connect people who are inspired and interested to be able to do things for patients that don't otherwise have an effective therapy or treatment. And I think it's such a unique opportunity to be able to see the patients as we do in the clinic or in the hospital, and then be able to apply the research techniques that we've learned how to use, and then be able to use our collaborations with other researchers who don't not necessarily have the same contact with patients, but have a really incredible skill set or inspiration to be able to bring new therapies for patients who don't have otherwise effective treatment. So that's an inspiring story that your research has been able to tell. So so exciting to talk with you.
Thanks. Thank you so much, David.
Should we wrap up with our traditional end of the show question, which is how do you and your team relax? What do you do when you're not investigating cutting edge prenatal therapies?
Well, one of the traditions we have is our weekly tradition, which is we start lab meeting with rosebud thorn. And so everybody, we go around the room. What's the best thing that happened for you last week? What's the worst thing that happened? And what are you looking forward to? And it becomes a really nice check in about, well, what are people bringing to work that they would like to talk about? And, especially the roses are amazing because obviously there's, life celebrations. One of my grad students got married over the weekend. But then there are also, challenging situations where oh, that experiment didn't get done because this was happening in this person's life and what we look forward to. So I started doing that with my children 10 years ago, and it sort of improved the dinner table conversation. And I feel like it's been a really rewarding um, tradition we have in the lab now.
Can I say one other thing that I know about Tippi? Because I walk into her office almost every day, is that she has a piano in her office. Tippi is also a Juilliard trained pianist, and she has headphones like this. And so there's also, to give a little bit of personal insight, she has this beautiful piano that she has invited my children to sit and play at when, we're juggling different things. And so there's also some beautiful music that takes place in her office and, around the Stem Cell Institute, thanks to her classical music training. Alrighty. Well, thank you so much, Dr. MacKenzie, for being with us today, for sharing your insights on, really prenatal cures, which I think, should be hopefully an important tool in our toolbox moving forward. And it's been beautiful to imagine this brave new world with you. David, anything further?
No, that was great. It was so great to see you, Tiffy. Your work is so inspiring, and I think among the neonatology physician scientists that are listening to this and our trainees who are inspired to do work like you're doing, it's so empowering to hear, how you built your career, what inspired you early on, and the questions you're asking now, and how you've been able to do the work that you're doing. So, thank you so much for taking time to talk with us. We really appreciate it. And know it's time for your lab meeting. So,
And David, I really appreciate the invitation and thank you for having this podcast. It's a really wonderful effort. Really appreciate the privilege of being on. Thank you.
excellent. Thank you so much, Betsy, and thanks to our audience for tuning in. And we will be back in the new year with a new interview. So thank you so much and have a good day.
Stay tuned for more. Take care. Bye.